Hi. So as we learned last time, the hippocampus has a lot of glutamatergic synapses where plasticity can happen. Um, these now there are lots of different glutamate receptors, but the two ionotropic ones are the AMPA receptor and the NMDA receptor, and those are both at the Schaefer collateral synapse. Okay, how do they contribute to plasticity? They have very different roles, and so this video is all about clarifying those roles. So as I said earlier, right, the presynaptic terminal releases glutamate. On the other side, there's AMPA receptors and NMDA receptors. How do these work together to make the synapse function and enable it to do plasticity like LTP or LTD? So I think, though, that based on what you've learned so far, you can start to address this question in your head. So imagine this synapse, right? A synapse like this one that has both AMPA receptors and NMDA receptors. When will these receptors pass ion? All right, well, basically, whenever the glutamate is released, AMPA receptors will open every single time. It's only glutamate gated. However, NMDA receptors will only open if the synapse fired just recently. Most of the time, if you recall, the NMDA receptor is voltage gated. It has that magnesium ion that is blocking the pore, and that will only be kicked out if the postsynaptic membrane is depolarized. Most of the time, the membrane is hyperpolarized, so the NMDARs can't pass ion. So this includes at rest. Only the AMPA receptors will open. But if a synapse has just been stimulated with a stimulation protocol, the membrane will still be polarized from the previous. So like you do one, then the membrane will still be depolarized from the previous firing event. So that magnesium ion will be kicked out and the NMDA receptor can open and pass ions through if there's glutamate. So given that, which ionotropic glutamate receptor determines the amplitude of the normal EPSP and which one is the coincidence detector? Well, all right. It's actually C. The AMPA receptor determines the amplitude of the normal EPSP, but the NMDA receptor is the coincidence detector. Why? So we said that the AMPA receptors open every single time, and even when the neuron is at rest. So most of the time, right, you're not going to get like 100. You're not going to get like 100 stimulations a second. You're going to get like one every once in a while. And so when measuring the EPSP amplitude, just the raw one, like all these dots, the cell is stimulated infrequently. Okay, so you're just gonna do one every like couple seconds, maybe, or even every like minute or something. So you do it infrequently enough that the postsynaptic membrane should still be at rest in between. And when you do that, only the AMPA receptors will be activated. So your AMPA receptors give you kind of your normal workaday. Okay, I have stimulation and I am getting an EPSP on the other side. And so for that, that is only going to be carried by the AMPA receptors because the NMDA receptors don't even open when the postsynaptic cell is at rest. Okay, so everything you see here, this whole plot, all of these dots, those are measuring basically AMPA receptor current, or sorry, AMPA receptor mediated PSPs. Okay, now what about the coincidence detection? So if you remember, what is coincidence detection? Coincidence detection is when the presynaptic cell fires, the postsynaptic cell fires, and there's something the detector can tell when both of those things are happening at the same time. So let's think about whether the NMDA receptor might be a coincidence detector. So we said that the NMDA receptor, it's only going to open when glutamate is released, right? That's when the presynaptic cell is firing. And when the postsynaptic cell is depolarized for a sustained amount of time. That is indicative of postsynaptic cell firing. So that's the coincidence. The glutamate release and the postsynaptic depolarization, that is the coincidence because it's two things are happening, the presynaptic cell firing, the postsynaptic cell firing at the same time. And the NMDA receptor will only open, right, when both of those things are happening. So it is functioning as the coincidence detector because it's only opening and letting in calcium when both those conditions are fulfilled, and both those conditions are fulfilled only when both neurons are firing. So our NMDA receptor, that is our coincidence detector. So question for you, in this experiment, a control slice of hippocampus was stimulated twice to get LTP twice, okay? 1 and 2. So the stimulation happened here and then here. So if you did that with a slice of, and when I say slice, I mean like somebody cut up a mouse hippocampus or a rat hippocampus and was like doing, looking at LTP in a dish. Okay, if you added APV, what would that look like? And APV blocks NMDA receptors. All right, well, we said that that our NMDA receptor was the coincidence detector, and it is where all the LTP starts, late or early, that's where it starts. And so without it, it's going to stay at baseline. You're going to get short-term potentiation, but that's going to decay within minutes. And otherwise, you're going to stay at baseline. What would this look like if you blocked AMPA receptors? Well, if you blocked AMPA receptors, you wouldn't be able to measure anything. All of these dots represent a potential that is coming through AMPA receptors. So if you blocked AMPA receptors, you wouldn't get anything. So looking back, we talked about how applying APV to mouses or rat hippocampus, it blocks spatial memory formation. So if you recall, these are the probe trials from the water maze, and it's showing that if you applied APV during training, the rat just like goes wherever. It doesn't actually know where the platform is. It doesn't spend its time near the platform searching for it, right? In the probe trial, the platform isn't there. The control rat, like, at least looks for it where it's supposed to be. And then the APV given rat, like, just goes around and around. And so why does it make sense? So again, the APV is blocking the NMDA receptor, which is doing that coincidence detection. So without that coincidence detection, LTP isn't happening, that synaptic plasticity isn't happening, and this is suggesting that it's blocking the, so APV should be over here, it's blocking the memory formation as well. All right, so I hope that makes sense, and that it also makes sense now why we spent so much time talking about the NMDA and AMPA receptors. It's because these two things are functioning very differently. The AMPA receptor is responsible for like moment-to-moment EPSPs, whereas that NMDA receptor is that coincidence detector that's really, really important for plasticity and learning and memory to happen. All right, see you next time.